What we're going to look at today is to see how the DNA mRNA code works. And so the purpose of this is to see how a particular protein can fold up into a particular shape to give a particular function and how that's directed by the DNA sequence. And this is going to be really important because it's going to help us understand how mutations work. I'll show you a quick example of that at the end. And to see how you can get different traits from just changing one base of the DNA sequence and how that ripples through the mRNA to the amino acid sequence of the protein and to the trait. So to begin with, what you're going to see here is that I have a DNA sequence here, TAC, et cetera, et cetera. And so let's say that this represents our gene. Well, according to the central dogma, we don't actually use the DNA to make the trait to guide the production of the protein. So the DNA will, through a process called transcription, make a copy of itself as mRNA. So that is the first thing that we're going to need to do. So we're going to make the mRNA. Remember, the mRNA has a U instead of a T, but the T in the DNA still pairs with an A. It's the A in the DNA that pairs with a U. So we just look at our sequence of the DNA. Let's be careful. We don't want to make a mistake. And CCG. And there is our mRNA. It will go to a ribosome. Well, what will happen in the ribosome is the protein gets made. How do we make the protein? Put bonding one amino acid to another. But which amino acid? How does the cell know which order to put them in? There are 20 different amino acids. So that is the question. How does this mRNA guide the production? Well, the key, as we're going to see in tomorrow's assignment, is that there are going to be tRNAs. And what the tRNAs do is they're able to bond with the mRNA in groups of 3 bases. So every 3 bases in the mRNA is going to represent a particular amino acid. And each of these 3 bases in the mRNA is called a codon. So the first codon in this mRNA, I drew a green line there, is AUG. And so this is codon 1, AUG. Codon 2 is the next 3 letters. Here we go, CCU. So that is codon 2. The next codon, you take a look, it would be GAU. The fourth, UGA, and the fifth, CCG. And I do like when I'm looking at the mRNA and I'm doing a coding problem, I do like to draw these little green lines every 3 bases. If I don't do that, I'll lose my place, and I might end up either skipping a letter or using a letter twice. So drawing that line helps me as I go. So the idea here is we've got this first codon. Okay, here we go, AUG. What that does, those letters, AUG, it tells the ribosome through the tRNA what amino acid goes into the first position. And you'll see down here, I have a kind of a start of a protein. Each of those ovals representing an amino acid. And what I'm going to do is put the name, or at least an abbreviation for the name of the amino acid that this mRNA is guiding. So how do I determine that? The first codon, AUG. I'm going to come over to this little chart over here. This is called an mRNA codon chart. And what it's going to do is it's a code. It's a universal code. Every organism uses the code. So whatever AUG amino acid is in a human, it's the same in bacteria, in yeast, and plants, and other animals. All uses. So this is why you can take a gene from one organism, put it in another, and it still functions because each animal, each organism can interpret the gene the way that it was intended because we all read the code the same. So you never have to memorize this chart. You'll always have this chart available when working on any problems, any tests, anything like that. So here we go. First codon is AUG. How do I read this chart? I read it from inside out. You'll see that there is this first circle here, kind of shading it in. That has the big font letters, a G, an A, a U, and a C. Okay, so that's it. The first codon, first base in the first codon is A. So I'm going to be focusing on this one right here. This is my first codon base A. And then I'm going to go to the next ring. So that's going to take me down to here. I see there's a U, and my third base in this codon is G. So here it is, A, U, G. I just kind of read this going out from inside out. What is the first amino acid? It is called methionine. All these with end in ine, those are pronounced ene. So methionine, and you'll see that I gave you the three-letter abbreviation. So what is the first amino acid in the polypeptide for this gene? It is methionine. So I'll write MET right here. Okay, what is my second one? You might pause, see if you can figure it out. Give you a chance to do that. Okay, so what do? I look up what's my next amino acid. I'm going to look up codon 2, which is CCU. That's going to start over here, C. I'm going to go here to C. And here you'll notice this is very common. The third base in the codon did not really matter because as long as the first two bases are C and C, the third base always gives you the amino acid proline. So proline will go here. And my third amino acid, again, pause if you want to try to figure it out. I'm going to look up the codon GAU. So GAU, here it is. And you will see that that is the amino acid aspartic acid, and its abbreviation is ASP. So there's my first three amino acids. I've now completed that. So why is there not another amino acid if there's a fourth codon? Let's look up UGA. So I have, here's the U, here's the G, and here's the A, and oops, there is no amino acid. It tells me to stop. What do I do when it says stop? I stop. I stop. What does that mean for codon 5? It means I ignore it. And this is, in reality, this is what happens in the cell at the end of the coding part of the mRNA. There will be a stop codon. Instead of an amino acid going into place, a release factor will come in and the polypeptide will be released, and that will be the polypeptide. This is obviously an example in real life. The amino acid chain sequences or polypeptides are much longer than three amino acids, but this will work for a little demonstration. So I'm done. I have figured out the amino acid sequence for this protein. And if this were a real protein, it would fold up based on the properties of the amino acids. Remember, they each have an R group, and those R groups give each amino acid a different property. So either some are ionic, so there's some that have positive R groups, some that have negative R groups. And then there's some that are hydrophilic, and then there's some that are hydrophobic. And so what can happen is that if you change the DNA sequence. So imagine that this second G was turned to a C. Well, what would that mean? That would mean the mRNA would have a G in the second codon. Instead of looking up CCU, I need to look up CGU, and I wouldn't have proline here. Proline would be gone, and instead substituted for it would be arginine with its ARG. And so arginine would have different properties than protein, and that particular protein would fold up differently. So that is how a mutation, such as that change from G to C, would end up causing a different trait. It puts a different amino acid in place. So hopefully this gives you a feel for how to complete coding problems. This is a very important part of biology. And so if you have any question, please let me know. Bye.